Okay, I think we probably ought to get started. My name is Abul Abbas, and I'm one of the faculty for this course. And before I start with the course itself, a couple of general sort of comments. But before I even do that, let's take a show of hands. How many people in this room are from biotech or pharma companies? Well, fair number, almost half. How many are academic researchers, lab researchers? And how many are fellows or students? Minority. So about half the group is. And so one of the challenges that creates, of course, is that this is a very diverse audience, and you guys have different levels of expertise, and some of you have done immunology courses recently, some have done immunology courses a long time ago. And so it's always a challenge to teach a course with such a diverse group. You know, if you think back to medical school or graduate school, it's not so much diversity. Most of the students come in with more or less similar backgrounds, but this is an extremely heterogeneous group of people. So it does create challenges, so I'll tell you. And the second problem is that we have a limited amount of time, 6 or 7 hours. So it's pretty obvious it's a pretty obvious statement that we're not going to teach all of immunology in 6 or 7 hours. So what are we going to teach? So the way we're going to do this is to do a very little time on some very basic principles, and that's just intended to get everybody up to speed, just so you have a sense of where the field is at, but spend most of our time talking about what all of us find more interesting, which is what the recent advances are, and especially recent advances that have relevance to human disease and therapeutics. In order to do all of that, it doesn't work if the three of us who are the three faculty members speak at you and get nothing in return. And so it only works if you participate in the process, which means interrupt us, ask questions, challenge us, and so on. Now, those of you who've heard me teach in other forums know that I often say that if you don't ask me questions, I'm going to start asking you questions. And usually, my questions are much tougher than your questions. So as an exercise in self-preservation, you need to interrupt me and ask questions. It's also the reason why I ask the people who are finishing their breakfast back there to move up and sit with the group here. Partly I can make eye contact, but mostly because I can ask you guys difficult questions. So seriously, interrupt us. I also want to make one other small point, which is many of you have fairly specific interests. You're working on a particular project, a particular molecule, or a particular pathway, and I can't guarantee you that we are going to cover that molecule or pathway during what is really a survey course. It's an overview kind of course. So we may or may not be talking about what your specific interests are, and so it's really up to you to find us. Find us at breaks or during the rest of the meeting and ask us about whatever you might want to. But be interactive. If you lose me along the way or lose any of us along the way because we are not being clear, you'll lose the rest of the talk. So if you start losing us because you don't get something, be sure to stop us and ask. Okay, that's the preamble. So what are we going to do? And by the way, this 2-slide business is really, I find it really annoying. I'm not going to be communing with my laptop and sitting there moving a cursor around because I like walking around. So the price I pay for walking around is that I have to point to 2. And that's only a sort of small problem because this light is so weak that even pointing to 1, you can't see anything anyway. So pointing to 2 is about just as bad. Andy, maybe you can find a, yeah, something that's a little bit better than this. Because you really can't see that, can you? Okay, so what are we going to do in this course? So we're going to emphasize nomenclature because immunologists have their peculiar jargon, and we kind of use words that nobody else can understand. Sometimes other immunologists don't understand what you're talking about, so it's important to appreciate the language, the vocabulary of immunology. But I'm not going to spend, or we're not going to spend a lot of time on that. Little bit on basic principles, just the nuts and bolts to get us started, but really much of the focus will be on emerging concepts and what their potential implications are. And so I want to start by just asking a very simple question. Why is the world out there so interested in immunology? And this is not just the medical and scientific community, but you can actually pick up newspapers and you'll see articles on the immune system. So why are scientists, physicians, and even the lay public so interested in this field? And there are many answers. As a scientist, I think, obviously with a little bit of bias, I think that if your goal as a scientist is to study and understand a complex biological system, as a biomedical scientist, this is a glorious example of how well we've done it. We've taken it apart and we are starting to put it back together again. So as you're going to see during this course, I can tell you when a virus enters your skin, I can tell you what cells capture that virus, where those cells go, how they display the viral antigens, who recognizes those antigens, how the cells that recognize them come back and get rid of the virus. This is a multiple-day, multicellular process, and I can tell you all the molecules involved. And that's a pretty impressive statement, that you actually understand all the components of a normal immune response. And so that's been the real success, one of the real successes of immunology. But its interest to a lot of us is that it is the cause of many different diseases, and it likely, the immune system, when I say it, the immune system likely impacts many more diseases. Even things that you don't think of as immunological diseases have some interaction with the immune system, and we're going to touch upon some examples of those as we go along. But I think in the last 10 or 15 years, the really impressive, yeah, much better, thank you. The really impressive accomplishment of the field is this third point, that if as scientists your goal is to use fundamental knowledge to develop new therapies that will impact human disease, I would suggest that the immunologists so far have done that better than anybody else. We have changed the course, we as a community have changed the course of many immunological diseases by developing therapies that are absolutely based on rational principles. And this is one of the most glorious successes of the field, not just the basic science, but its applications, implications to biology. And then the last point I want to mention is that these therapies are targeting immunological diseases like rheumatoid arthritis and Crohn's disease and psoriasis and so on. But it's now understood that, that the immune system, or manipulating the immune system, may have a major role in many other therapies. For example, if there's all this excitement about regenerative medicine. If you take embryonal stem cells and inject them into an individual just systemically to try and repair that individual's heart or something else, what will happen to those ES cells? What will happen to those embryonal stem cells? Anybody? So the result of immunizing will be that those cells are going to be rejected. There is no practical way of matching ES cells with every potential patient out there. So if you use ES cell-based systemic therapy, the cells you inject are certain to be rejected. This regenerative medicine stuff is dead in the water until we figure out, at least some aspects of it, until we figure out how to manipulate the immune response. So the implications of immunology go way beyond rheumatoid arthritis and psoriasis and Crohn's disease. It has impact on, by the way, many of you know that systemic gene therapy protocols worldwide now are shut down. Every single one is shut down. It's shut down because of the adverse effects of the immune response to the viral vectors that were used to deliver the genes. So gene therapy is dead in the water until we can figure out what to do with the immune system. And so the potential for the immune system playing a role in major therapeutic strategies is enormous. And we all need to understand and therefore learn to manipulate our immune responses. Okay, so that's kind of a preamble. So what does the immune system do? We now know that the normal function of the immune system is defense against infection. That's what the term immunity means. Immunity means protection against infection. So that's about the only really useful thing the immune system does all the time. It also protects people from some tumors, and later on we're going to talk about the resurgence of interest in this idea of tumor immunity, which was a shaky field for a long time because it was difficult to prove that the immune system was doing anything against tumors. That idea has been completely reborn over the last few years. So those are the normal functions of the immune system, but it's also the cause of many diseases, allergic and autoimmune diseases. This course has no discussion of allergies. And at the end of the day, I'm going to talk about autoimmunity, but in very general terms. We're really not going to discuss any specific autoimmune diseases. So we won't talk about these diseases much. We will talk about general principles. I already mentioned that the immune response is the mechanism by which cell and tissue transplants are rejected. And I also mentioned this idea, which I think you'll hear a little bit about, that the immune response, and specifically the inflammatory response, is suspected to play a role in many diseases that you don't think of as immunological diseases. So it's suspected to play a role in type 2 diabetes and the metabolic syndrome, in Alzheimer's, in atherosclerosis, and many of these other diseases. So targeting inflammation in these non-immune diseases is becoming a big deal out in the community, in the scientific community now. So, how do you defend yourself against infection? And much of what we're going to talk about will be in that context. So the answer is that there are two groups of immune responses, two types of mechanisms. The early defense mechanisms are what we call innate immunity. I'm not going to go over any of these in any detail because Andy's next lecture will be on innate immunity, but innate immunity, remember, is what's present in you all the time. It's poised to fight against microbes. So it's always ready, always available. And it's what kicks in immediately if a bug enters you. So if a microbe enters you, your innate immune system responds right away. It controls the infection, it sometimes eradicates the infection, and it allows time for the more powerful mechanisms of adaptive immunity to develop. Adaptive immunity is called that because it's normally silent and it responds to infection. So it adapts to the presence of infection. This is what in the old days was called specific immunity. Lousy term because innate immunity also has specificity, or acquired immunity. Acquired meaning it developed after infection. It was acquired as a result of infection. modern nomenclature, we've abandoned all of that and we call them innate and adaptive. The adaptive immune system really consists of lymphocytes and their products, and the rest of this course, other than Andy's next lecture, will focus on mechanisms of adaptive immunity. This is very basic at this point. Stop me if there are any questions. So what are the major cells of the immune system? Lymphocytes are the only cells that have receptors for antigens that are highly specific, and I'm going to come to those receptors in just a minute. These are the cells of the adaptive immune system. They're the central cells of the adaptive immune system. But lymphocytes are not enough to get the response going. Antigens have to be captured and brought to the lymphocytes, and the cells that do that are called antigen-presenting cells. And after the coffee break, I'm going to tell you about antigen-presenting cells. And then this third cell population, it's a little bit of a loose term. So lymphocytes normally don't do anything. They just recognize antigen when they're sitting around. And once they've recognized antigen and been activated, they turn into cells that are capable of doing the job of the immune system. And the job of the immune system normally is to do what? The task of the immune system is to get rid of infection. So lymphocytes that see antigen turn into cells that actually get rid of the antigen, or recruit cells that get rid of the antigen. The cells that do the job, the cells that mediate the effect of the immune response, are loosely called effector cells. Some of these can be various white blood cells like neutrophils, macrophages. Those are all effector cells, and some of these can be lymphocytes. Plasma cells can be considered the effector cells of the B cell system, and I'm going to come to that in just a sec. So effector cells are the cells that are doing the job. And it's a somewhat sort of vague term, but it's a term that's used often enough that you should be aware of what it means. And so how do lymphocytes recognize antigens? We're not going to talk about how the receptor diversity is generated, but you should understand the principle. So it turns out that a lymphocyte precursor comes from the bone marrow and goes to the organ in which it matures. The organs in which mature lymphocytes are generated are called the generative lymphoid organs. And the generative lymphoid organ for T cells, which we're going to talk about in a sec, what's the generative lymphoid organ for T cells? The thymus. And what's the generative lymphoid organ for B cells? The bone marrow. So lymphocyte precursors either stay in the bone marrow or go to the thymus. And once they get to their appropriate site, they start generating lymphocytes with many different antigen receptors. I'm showing only 3. The best estimate we have is that there are 10 million. There are 10 million different antigen receptors generated. Now, the molecular mechanisms by which that happens are extremely well defined. Everybody knows how that happens. It's by rearrangement of antigen receptor genes, and we're not going to talk about it at all. But that's very basic textbook reading, so you can read about it yourself. So 10 million of these receptors are generated. Each cell has only one receptor. Therefore, each cell has only one specificity. Each cell and its progeny are known as a clone. So in the immune system, we think of clones as lymphocytes with single receptors and therefore single specificity. Antigen receptors are therefore said to be clonally distributed. They're different on different clones of lymphocytes. So these cells will be generated in the generative lymphoid organs. They'll wander off into the circulation, and they'll seed the peripheral lymphoid organs. And when a microbe enters, it interacts with a cell with predetermined receptors. So the cells are already sitting in you and I with predetermined receptors, and the lymphocyte now recognizes microbes that enter. The result of that recognition is that lymphocytes are activated, they proliferate, they differentiate into effector cells. In this case, this is the B cell lineage, and those effector cells start making antibody against whatever was being recognized. And so that's the principle of what was called 50 years ago the clonal selection idea, that clones of lymphocytes with predetermined receptors exist prior to encounter with antigen, that antigen selects the right clone and turns it on. And this is an absolutely fundamental concept, that you and I all have predetermined receptor specificities. So what are the lymphocytes we are talking about? B lymphocytes are the ones that go on to make antibody. And I'm not going to talk about these effector mechanisms. We'll touch upon them later on. T lymphocytes are divided into two main classes. Helper T lymphocytes work mainly by secreting cytokines. And of course, these cells can be distinguished by the expression of surface markers that are named according to the now unified CD nomenclature. So helper T lymphocytes express which CD molecule? Helper T lymphocytes express CD4, and killer T lymphocytes or cytotoxic T lymphocytes, which kill infected cells, of course, express CD8. So CD4 and CD8 distinguish helper T cells and killer T cells. We now realize that there is a third population of lymphocytes that neither helps nor kills, but whose main function is to suppress immune responses. And so we now consider regulatory T cells as an independent T cell lineage. It's got independent properties and functions. And in the afternoon, I'm going to tell you much more about regulatory T cells. We include natural killer cells here because natural killer cells are lymphocytes, so we're talking about lymphocytes. Are natural killer cells part of the innate immune system or the adaptive immune system? Innate or adaptive immune system? Innate immune system. They do not have clonally distributed receptors. They're poised to kill all the time. They don't need to be activated. So NK cells are part of the innate immune system, and in that respect should not be considered members of this group. We lump them in here just because, as I said, we are talking about lymphocytes. So how do these lymphocytes respond? And this is so simple, you don't need to go over it in detail. Humoral immunity is the kind of immunity that's present in your secretions. So in medieval medicine, there were 4 humors. What are the humors? What are the humors? Your secretions, your soluble stuff. Blood, bile. There was actually 3 humors. Blood, bile, and phlegm, sputum. And so the kind of immunity that's present in your serum and in your gastrointestinal and respiratory secretions is called humoral immunity. Remember, those were the days when you were sick because your humors were evil. And that's what led to the practice of bloodletting, right? You were getting rid of evil humors. So the term humoral immunity really rose from that historical context. And humoral immunity is mediated by antibodies, and it works against extracellular microbes. But if a microbe has been eaten up by a phagocyte, or if a microbe is infecting, a virus is infecting some cell that cannot get rid of it, like a hepatitis virus inside a hepatocyte, then these microbes are inaccessible to antibodies, and they're eliminated by the mechanisms of cell-mediated immunity, which are all mediated by T cells. So cell-mediated immunity is T-cell-mediated immunity. It gets rid of microbes within phagocytes, and it gets rid of microbes, specifically viruses, within non-phagocytic cells. It gets rid of things that are inside cells. Humoral immunity is antibody-mediated, and it gets rid of extracellular microbes. A couple of other very simple points. So a naive cell is the cell that's floating around in you and I, poised to recognize antigen. It hasn't seen antigen. Therefore, it is immunologically inexperienced. And when you were inexperienced, your parents told you you were naive. So that's how the nomenclature arose. It's an immunologically inexperienced cell that can recognize antigen. Once it recognizes antigen, that cell gets activated. Activation consists of two events. The cell proliferates, meaning that clone of lymphocytes of that specificity becomes a much bigger clone. So proliferation leads to clonal expansion, and then some of the progeny of that cell differentiate. Differentiation means a change in phenotype. So a recognizing cell becomes an effector cell. A B cell becomes a plasma cell. A helper T cell becomes a cytokine-producing effector cell. That's a change in phenotype and therefore in function, and that change is called differentiation. So the two events associated with lymphocyte activation are proliferation, leading to clonal expansion, and differentiation, leading to the acquisition of effector function. With me so far? It's really very simple at this stage. Okay, so that's kind of an introduction to some of the nomenclature of the physiologic immune response. I just want to end up with a couple of points. So this is just a repetition of what you probably already know. You know that the immune system can cause disease. But the point I want to emphasize is this third one, which is the realization. So, for 50 years, and this really started with Edward Jenner's vaccination, successful vaccination. For 50 plus years, immunologists have been studying how to turn the response on. How do you stimulate the response? That's what you do when you vaccinate someone. You stimulate a response. What we've learned in the last 10 or 15 years is that what's really critical in the immune system are the control mechanisms. That failure of control, so the reason why people get autoimmune disease or any inflammatory disease, it's not just that the response gets turned on, it's really that the control mechanisms have been disrupted. Something has gone wrong with control mechanisms. And so this idea has really become a dominant theme in immunology, that we need to understand the control mechanisms in the immune system if we are going to learn how to manipulate it better. And in my next lecture after the coffee break, I'll show you what is really the most brilliant example of understanding control mechanisms and exploiting them for therapy. So keep this idea in mind, that this is a dominant theme in immunology, understanding regulation and learning how to manipulate it. I want to end up with 2 or 3 slides on what some of the successes have been in very broad terms and what some of the challenges, continuing challenges are in the study of the immune system. So why are these recent advances in immunology important? Because it provides a fantastic foundation of basic principles, and you'll see that as we go along. Because the fundamental knowledge allows you to begin to understand disease mechanisms, and we are doing better and better at that. Also, this foundation allows you to develop novel therapies. So there have been these great advances, and I referred to some of these in an early slide. But what are the challenges facing the field going forward? And some of these challenges are not peculiar to immunology. The first one, and this is actually quite worrisome for me personally, but for many of you, is that as we collect more information, the first thing that happens is increased complexity. It really doesn't help you understand. It just builds up the information base, and it makes things much more complex. And this is true in the immune system in many different contexts. Just some examples. this whole population of cells, which I think Andy will refer to very briefly, called innate lymphoid cells. It's just flow cytometric identification of new cell populations. I'll refer to dendritic cells a little bit later. The number of subsets of dendritic cells is growing almost exponentially, and none of us understands whether that has any functional significance or not. The number of helper T cells. We thought life was simple with Th1 and Th2. Then the Th17s got added. We thought life was reasonably simple then. Now, everybody with any cytokine has their own helper T cell subset, and nobody understands what their significance is. So the first thing that happens when you have increasing information is actually increasing complexity. And that problem is compounded by a lot of the high-throughput omics approaches, genomics and proteomics. They're just piling up information. And you can read thousands of papers on genomic analysis of one pathway or another, and nobody knows what any of that means. So this is the challenge that all of us are facing as the technologies have come in. That doesn't mean it's bad to do the technology. That just means that we are at the cataloging stage, and it's going to take us some time to learn it. The second problem in immunology, and this is especially a problem for people studying humans, is that although we all believe this, and this is very strongly my belief, that the truth is in vivo and the culture stuff that we do is maybe. The problem, of course, is that in humans, all you can do is cell culture stuff. There is no way of doing an in vivo experiment. And so the result is that much of our knowledge, especially of the human immune system, is based on cell cultures, and it's very difficult to convert that to in vivo biology. And the problem is compounded by this, that if the emphasis, the emerging emphasis is on regulation, it's extremely tough to model regulation in a culture dish. That regulation is complex and there are multiple cell populations involved, and you just can't mimic it in a culture dish. So you really basically are stuck with studying it in vivo. So this is one of the big problems facing us. The third problem facing us, which all of us have become acutely aware of, is that although experimental models, specifically mice, have been spectacularly successful, they have taught us all the principles of immune responses and have been completely essential for discovery. We are also now realizing that they may not accurately reflect the human situation. And this is especially true when you get to immunological diseases. There is no mouse model, none, that mimics Crohn's disease. There are all these things that people try, and not a single one of them actually accurately reflects Crohn's disease. And the same is true of many other inflammatory diseases. And the result is that animal models of disease have not proved tremendously predictive. What astonishes me is, despite the poor predictability of animal models, it's astonishing how many successes we've had. Anti-TNF, anti-IL-17, all these drugs have been based exclusively on animal model experiments, and gutsy clinical trials really based on pretty lousy animal models. So we now understand that these models are imperfect. It's amazing that they still work, but going forward, all of us, people in pharma, people in academic institutions, are going to have to figure out how to solve this problem. And this is going to be one of the big challenges facing the community over the next decade or two. So I've tried to introduce you to some of the basic nomenclature. I've given you a sense that the field has been really exciting, but I also want to emphasize that there are lots of challenges facing us going forward. And I will now stop and let Andy take over.